Okay. Yeah, thanks again, Colin. So first off, thanks to all you for being here. Thanks to Colin and the Funga crew for inviting me. Yeah, as Colin said, we've known each other for a long time, so it's really nice to see Funga start to grow and see where it's coming from. So really happy to answer some questions as well afterwards and talk about this research and how it might fit in with some of the work you're doing. So yeah, I'm at University College London. I started in August last year, so relatively new. And I'm going to talk a little bit today, like I said, about everything that Colin talked about to some extent, and really focusing on this thread that unites a lot of my research, which is what I call diversity begets diversity, these idea of feedback loops that can help promote and promote and maintain diversity across scales. So I'm going to talk a little bit about experimental work with fungi, some theoretical work, and then go up to macro scales looking at global forest systems. So as we all know, unsurprisingly, natural communities are typified by very high biodiversity. And of course, when we look at tropical forests, we see this, thousands and thousands of species of plants and animals. But even when we look at areas that we might think of as having relatively poor biodiversity, so subarctic tundra, when we move below ground, we also see really astronomical levels of biodiversity, bacteria, fungi, arthropods, insects. And so across the globe, we really see almost every ecosystem has high biodiversity. Just the exact location might differ, above ground, below ground, soil, plants, and so on. There's a bit of a challenge, though. If we view biodiversity as this stacked rocks here, and what we want to do is stack up an ecosystem with a lot of biodiversity, traditionally people have turned to these very basic models to try to sort of understand and simulate dynamics. The challenge is when we use these models, we inevitably really struggle to build diverse ecosystems. We could maybe have 2 or 3 species, but inevitably they collapse into monocultures unless you really fine-tune these coefficients. So what people have spent the last 30 or 40 years doing is building more and more complex models where they add in things like niche partitioning across gradients and diffusion across space. And maybe with these more complex models, they can start to build more diverse ecosystems. But still, what we get out of these models is a really poor facsimile of what we see in nature. So there's this real fundamental challenge. And as a community ecologist, what I'm really interested in is what maintains biodiversity. And when we look at these first principles, quote, mechanistic models, we really struggle to recover this high level of biodiversity in these systems. There's a really nice paper by Nathan Kraft a few years ago that really illustrates this challenge. So they're looking at 18 species of grassland plants. This is in California. And these are 18 species that have coexisted, as far as we know, for over a century. So essentially what we would call stable coexistence of a community. And they did this classical ecological experiment where you compete all pairs of plants against each other and you see who beats who in competition. And then you can extract some of these variables and you can map out this sort of interface line. And anything where when you plot out these pairs of species, if their outcome turns out down here, they can coexist. If it's above this line, you get competitive exclusion. We can sort of ignore the mathematics. The key thing I want to draw your attention to is just that out of all of these pairs of species, only a tiny fraction would be predicted to coexist. The vast majority of pairs of species, one would quickly displace the other. This creates this real challenge because this is using sort of the cutting edge first principles community ecology, and we fundamentally fail to explain coexistence in this system where we know these species are coexisting. They go on to sort of give some examples, say if we bed them in more diverse model communities, and then say maybe it's related to things like multi-species competition, including intransitive competitive networks. So really what they're saying here is that coexistence, stability, and biodiversity more broadly is an emergent phenomena, and that we can't really get there by building these little models and understanding pairs and hoping to scale up to the whole. Rather, the properties of that whole system are going to emerge only when we consider all species simultaneously. So this is where a lot of my early work focused on. And what you're looking at here on the right is a model experimental system I have. These are wood decay fungi, so same fungi that produce mushrooms and dead wood. And you're looking at 2 species fighting against each other on a petri dish. So each petri dish has 3 little plugs of an isolate, and they grow towards each other and they meet and they have this sort of complex interaction zone. So we had over 20 different species, over 40 different isolates, so multiple phenotypes of the same species. And the question was, can we start to take what I call network approaches? So rather than just try to mathematically, look at individuals and then sum up to the whole, can we instead look at the entire network of interactions and try to make some broad inference about biodiversity? And again, these are really, really complex the way they interact. They sort of leach enzymes through the substrate. They're trying to decompose each other. They're using volatiles to try to kill off and inhibit growth of each other. So we know competition is incredibly complex. But what if we just reduce these interactions to what we call networks? So this network just shows who beats who in pairwise competition. So again, sort of mirroring what Nathan Kraft did. But the question is, now that we have this network, can we start to summarize biodiversity just by summarizing a property of this entire network? So we competed all of these isolates against each other, more than 600 different competitions. And we can see there's a clear best competitor up here, wins in about 75% of its competitions, and a clear worst competitor that loses in about 75% and draws in about the other 25%. So somewhat hierarchical, but also very complex network. Even the best competitor wins or loses and draws against weaker competitors as well. So the first thing we looked at is this idea of traits. And a lot of what I'm going to talk about is this through the lens of functional traits. Functional traits are really powerful because they give us a way to place species in a continuum in relation to other species by understanding their sets of functional traits. And for fungi, that's things like enzyme production, hyphal density, how fast they grow, decomposition rate, these sorts of things. When we look at pairwise competitions, when we look at the results of these pairwise competition, we see that the results are what we might call hierarchical, or they appear hierarchical. There's essentially a best set of traits and a worst set of traits. You want to be a fast grower, you want to have high density, you want to be very metabolically active, produce a lot of enzymes, decompose very heavily. And we can sort of think of this like gladiators in an arena. If we put professional gladiators in a Roman arena and we tried to join, we don't have the traits to survive. If you let this massive combat play out for a day, the only ones that would even have a chance of surviving are going to have very similar traits. And that's what we see in this system. So based on pairwise interactions, we would say, okay, competition is going to drastically reduce the diversity. The only thing that's going to survive if we embed them in more diverse models is going to be the few that look like the perfect gladiator in the system. So we can actually go and do that. So we can sample species from our network. We can place them in these big 30-centimeter Petri dishes. We did up to 12 species. We can also use sort of basic models like the zero-sum replicator equation to generate theoretical expectations. And then we can track the dynamics of these communities over time to try to understand how these pairwise relationships scale up to multi-species competitions. And what we find is that simple network properties can explain the vast majority of coexistence. And in particular, what Colin mentioned, one of my favorite keywords, this word intransitivity, can explain almost half of what we see in these multi-species communities. So intransitivity is essentially a measure of how hierarchical a system is. And a hierarchical system is there's a best competitor and a weakest competitor, and the best beats the ones below it, the one below that beat the one below that. So A beats B, B beats C, A beats C. So you can rank them from best to worst. A detransitive community with 3 species we call a rock, paper, scissors community. So in pairwise competition, every species wins and loses against another. So rock beats scissors, scissor beats paper, paper beats rock. But when you embed them in this 3-species system, there's now no one best strategy. Every species has a winner and a loser that they can defeat against. We can take these rock, paper, scissors communities, and we can actually generalize this idea of intransitivity to as many species as we want. And what we see is when we build these more diverse communities, once you pass this threshold of about 50% intransitivity, meaning about 50% of the species are involved in an intransitive loop, we have almost total coexistence of all species in that system. And this is just one variable, so the intransitivity is really common, but we can look at things like nestedness and modularity and motif counts, and these also account for some of that residual variation in coexistence. What's really powerful about this is we can get at some idea of whether species can coexist without having to know anything about growth rates, anything about enzyme production, spore production. Simply by understanding the shape of this network, we can start to make some of this inference. Going to the title of the talk, I think what's most important is that it really shows this idea of diversity begets diversity. So what we have here is the phylogenetic diversity and the functional diversity of these communities as we increase the initial richness of the system. And what we see is that we increase the initial richness, we actually retain most of what we started with. So if we only have 2 species, we lose about 50%, unsurprisingly, because one species tends to go extinct. But as we get up to about 10 species, we're only losing upwards of maybe 15 to 20%. And you can imagine a piece of wood can have 100, 500 different wood decay fungi in it. So if we extended this curve, we're going to retain almost every single species somewhere in that log, which means we're going to retain most of that initial functional and phylogenetic diversity. What's happening here is the more species you add, the less these pairwise relationships matter. So as you add more and more species, you break down the importance of pairwise relationships. What starts to matter is these emergent relationships between groups of species, and particularly these intransitive relationships, which breaks down any sort of destabilizing effect. So we call this diversity begets diversity, and I think there's a lot of people over the last 5 or 10 years, people like Santiago Salaveras, who've done really great work with intransitivity. And I think people now in the community ecology field really realize that it's probably a core mechanism that helps maintain high levels of diversity, at least over short time scales. And we can sort of see what's playing out. So if we look at maybe an example here, this is a 3-species petri dish, and you can see there's this one strong competitor. So low diversity communities collapse. Even if you have some degree of intransitivity, you still allow one species to generally sort of plow through. Once we have more diverse communities, that same really competitive species, maybe it's able to overtake 1 or 2 of its neighbors, but eventually it will bump into a set of species that it either loses against or that it deadlocks against. And this otherwise weak, really inferior competitor over here, which maybe can't really survive against any of these, happens to be insulated by the few that it can beat or deadlock with. And so this high diversity communities, these pairwise interactions play almost no role anymore because they're embedded in this spatial structure. So this can help explain why if you go and sample soil, if you sample wood, you're going to find enormous levels of biodiversity, much higher than we can explain, in part because these sort of pairwise relationships play very little role, and it becomes this emergent complex phenomenon. Now, there's a bit of the devil in the details. So as I mentioned, intransitivity, I think, is, a lot of community ecologists will bring this up in a PhD defense or something. What about intransitivity? But there's also something that people don't realize, which is that it doesn't provide stable coexistence. And when we think of things like those plant species coexisting, coexistence requires that oscillations dampen over time, or else variation will drive the ecosystem, drive the community extinct. So when we look at the dynamics of these rock, paper, scissors species, you get these really artificial, unbiological oscillations. So it's like, rock becomes dominant, then paper becomes dominant, then scissor becomes dominant. First off, we never ever see this in nature. Nothing looks like this. Maybe predator-prey dynamics track this a little bit, but not multi-species communities. So we know there's something fundamentally wrong with our model. More problematically, as soon as we add in any sort of variation, whether it's demographic variation, environmental variation, or just stochastic noise, these dynamics are incredibly fragile. They have this resonance in that system. And once you perturb them, that resonance magnifies over time until it sort of becomes chaotic and one or more species hits the boundary. And inevitably, no matter how you run these simulations, over long enough time periods, always a single species survives. So intransitivity can slow that rate of extinction, but it can't solve that problem of extinction. So we sort of set out to ask what might sort of prevent the system from exhibiting these oscillations through time. Now, in the system, as I mentioned, we have multiple isolates. So here you're looking at 4 different phenotypes of the same species. So these were cultured from across the United States. This comes from the US Forest Service Forest Products Laboratory in Madison, Wisconsin. And under any given condition, only one of these isolates is the most fit. Only one of them grows the fastest, would have the highest spore production. So at any point in time, maybe this upper right is the most fit phenotype for the agar it's growing on it, and the other 3 would be suboptimal. Now, from a population perspective, these other 3 might provide really important values. So things like bet hedging. So this idea that maybe right now they're not best, but in the winter, maybe they grow better if there's climate change or if there's a flood, right? There might be other conditions that they thrive under, which allows that population to be sustained over time. But still, at any one point, they're essentially going to be like friction on that system. They're going to be a detriment to the population level fitness. And so if you could essentially get rid of them, then maybe you'd perform better than having them included. But of course, they're linked to you genetically, so there's no way to get rid of them. So the challenge is, or the question was, can we start to model how phenotypic variation or genotypic variation would affect these dynamics? It turns out that no one had really done this before. So the question is, can we view a network? We still have that network of species. Each little circle represents a species, but we can actually group them together into different phenotypes within a species. So here, the green species is still just one phenotype, but now the orange species has 2 different phenotypes or genotypes. The blue has 3, the pink has 4. And the question is, how can we start to model the dynamics and make hypotheses about this system using this type of approach? So what we first had to do was derive what the model looked like. And we actually get this really nice, elegant looking equation, which just involves sort of two matrices, this Q and H matrix, which I'm happy to talk about, but I don't want to go into too much math here. Sort of the key result is that we actually can show through a change of variable that this is identical to this replicator mutator equation. Now, this is actually a famous equation in population genetics, and it describes how sort of alleles competing for frequency within a gene. And we show that this exact same model determines the dynamics of phenotypes nested within species. And the power here is it gives us an enormous amount of sort of mathematical machinery behind the scenes to focus on. We can then use this model to simulate dynamics. And again, this is purely theoretical. So the idea is what are the expectations for how this would affect the dynamics? So here we still have a rock, paper, scissors community, these light colors right here. You can see 3 species forming this sort of cyclical relationship. But every phenotype, every species now has a maladapted phenotype in dark that loses against everything else. And when we make this slight change to the system, we now see that the dynamics are stable. And essentially, this maladapted phenotype is a friction. It prevents those curves from reaching their maximum every time. And so it's sort of dragging down that population level growth rate because it's the average of a real fit phenotype and the average of a maladapted phenotype. And this stabilizes those dynamics. It prevents those oscillations from getting out of control. We can track what's happening here too. So we have this maladapted phenotype just sitting barely above extinction level, propped up by the fitness of the more fit phenotype, which converges to its equilibrium over time. We can do this with all different structures. So we don't just have to do rock, paper, scissors. Here we have 3 species where 2 of them only have 1 species and 1 of them has 2 phenotypes. And in this case, we still see stability. And again, you can track that maladapted phenotype. Now, this isn't just us playing games with these. We can actually show mathematically that this has to be the case. And this is some really great work by Jose Capitan, who's a physicist. And we're able to prove that when you add a phenotype into these dynamics, the dynamics become globally stable now. So this is a really powerful result. Again, just using network approaches, we can start to understand how phenotypic variation might affect the diversity of that system. And more importantly, what we show is that when you add demographic variation now, so whether if species grow at different rates, die at different rates, reproduce at different rates, or if we add in some sort of environmental noise, we now have stable dynamics provided at least one species has multiple phenotypes. And we also get this really nice result that the greater the difference between 2 phenotypes, the more stable that system is. So if species are really, really similar, essentially they act almost as one species and we get these oscillating dynamics. But as they get further and further and further apart, you're going to more quickly stabilize those dynamics. Again, this matches what we see from evolutionary theory, population genetics, that the role of phenotypic and genotypic variation is only really seen as a benefit when things are very different, right? There's no value in having a genotype or phenotype that's almost exactly the same. And we see the same result plays out at the community level as well. So these first 2 components, the sort of experimentally and theoretically, both iterate this idea from slightly different angles. So diversity begets diversity first, just higher initial richness can prevent extinctions sort of by breaking down pairwise relationship, by promoting intransitive loops. But then I think maybe more elegantly or beautifully, that variation at the smallest scale, so diversity within a species, is sort of fundamental to promoting community level stability. So it can help promote diversity at the ecosystem level. Now, of course, this is just one mechanism, right? We know that stability isn't just governed by intraspecific variation, for example, but it can help explain why we don't see these really chaotic oscillations that we might otherwise expect using just intransitive relationships. So that's sort of the first half. And now a lot of what my work on over the last few years has been to take a larger view. Say, okay, we have these first principles from experimental systems, from theoretical work. And this is great, and I still do a lot of this stuff. But at the same time, when we think of questions about climate change or non-native species, this is really about intact ecosystems. So as much now what I'm focused on is how can we use these sorts of results to inform broad-scale patterns, how we might start to manage ecosystems. So do we actually see evidence for diversity begetting diversity in real ecosystems? This is challenging, and particularly what I focus on mostly are forested ecosystems. And as many of you know, forests can take hundreds of years to mature. Trees can take decades and decades to grow, even to sort of beyond the sapling stage. So it's not something we can go and experimentally test. We have to ask these questions by essentially looking at broad-scale data sets that allow us to tease apart some of these signals. A lot of what the work I use revolves around this GFBI data set. So this is the Global Forest Biodiversity Initiative. Many of you might be familiar with this. It's essentially a compendium of country-level forest inventory data. So for example, the US forest inventory analysis, Canada, many countries across Europe, but also over 200 researchers have contributed individual plots around the globe. So it results in about 30 million different plots around the globe, giving us really high-level quality data about tree height, size, growth, and so on. So what you're looking at here is from a paper. This was by Camille Delvaux, who was a postdoc at ETH when Colin and I were there. She's now a group leader at ETH. And so we set out to sort of ask this question about what is the role of biodiversity in terms of invasion? And do we see some evidence for diversity begetting diversity or sort of insulating against loss of diversity? So this is what we call in invasion literature the biotic resistance hypothesis. So it's this hypothesis that does higher biodiversity sort of insulate or buffer a community against invasion and subsequently against further loss of biodiversity? So this biotic resistance hypothesis really is a question about does diversity beget diversity through the lens of invasion? Now what you're seeing here, all the plots, and the blue shows the intensity, the percent of stems in that plot that were non-native trees. And you can see there's sort of these hot spots around central United States and into Northern Europe. And when we look across the globe, when we annualize it to about a 50-year time window, we see about a 10% reduction in native diversity due to invasion. So aligning with what we all sort of expect, that when you have non-native invasive species, you either sort of, because species are physically excluded, or sometimes there's just not enough space, but you lose native diversity with the introduction of non-native species. And maybe 10% isn't that much, but this can amount to hundreds of trees in different regions. So it can have real strong ecological and as well as economic consequences. So we looked at two different things. And the first thing we asked was what's driving this spread? And there's sort of two ways we can do this. One is establishment and one is actual spread. Now, establishment is whether or not a species just gets there, right? So if we just look at a plot, is there at least one stem in that plot? So going up to here, at least one stem within this plot that is non-native. So just a binary prediction. And what we see is that establishment, whether or not there's a non-native species in a plot. And again, we're just talking trees, we're not talking plants. This is really driven by human activity. So establishment is a byproduct of humans. And this really makes sense because we're the ones bringing trees around the globe. And what we find in particular is that distance to ports is the strongest predictor of whether or not a plot is invaded or not by non-native species. And distance to ports is important for a couple of reasons. First off, just when you're close to a port, this is really the entry point for a lot of non-native species. So there's a mechanistic reason why this is the case. You know, seeds hitch a ride on shipping containers or planes, and then they get released into the wild from there. But also, distance to ports is a really strong proxy for human footprint. So if we think of really big shipping ports around the world, New York City, London, for example, these just have enormous population density as well. So distance to ports emerge as sort of the strongest human-centric predictor because it has a mechanistic and a correlative relationship. And, you know, looking around in London, for example, where I am, there's street trees everywhere. There's all these ornamental trees from Asia, and these things have a small but non-significant probability of escaping into the wild. But as you notice by looking at this, we actually see that functional diversity, and here it's decomposed into two different components. So the diversity through the lens of richness, the range of traits that species exhibit, and functional diversity through the lens of uniqueness. So how unique is any individual species? Neither of these have a very strong predictive power. So distance to ports maybe has at most about a 6% increase in invasion. And these really vary between 1% and 2%. You have to really squint to even see a relationship here. And in fact, we really see very little role of native biodiversity in terms of establishment. However, when we look at spread, which is after a tree becomes established, its subsequent ability to grow. So this is essentially the density that a non-native species attains. We now see that spread is a biological result. So spread is still slightly driven by distance to ports. We see a similar sort of trend, but we see this incredibly strong and very steep relationship with functional diversity, particularly functional richness, so the range of traits within a community, but also functional divergence as well. And so when you go up to the highest levels of functional richness, you have almost a 20% reduction in the density of non-native species. Conversely, when you have very, very low functional richness, you can have up to a 20% increase in the density of non-native species. So this really shows how the biotic resistance is operating. Biotic resistance isn't operating through the idea of establishment. That's driven by humans. Rather, it's operating by the ability to sort of buffer and prevent subsequent invasion and spread of that species. And we really see it's driven by functional biodiversity, so how unique species are, and how many, sort of their overall trait range that they occupy, with human activity still playing an important role, but not playing a hugely significant role. When we look at what this means across the globe, we see that high biodiversity can reduce the loss of species from about 10% on average down to about 1%. So in these real high biodiversity areas, we still have a slight loss of species. I mean, there's sort of no way to prevent that on average across the globe, but it sort of mirrors what we see in the experimental results, right? So when you increase your richness to these really high levels, really high functional diversity, for example, you still have a slight decline in richness. Species still inevitably go extinct, but we can recover much of what we would have lost in low biodiversity settings when you have very low functional phylogenetic diversity in these systems. So it's a really nice example where we can show how diversity begets diversity, and this actually helps insulate these systems by providing this biotic resistance and also getting some idea of where that biotic resistance is important, which can help feed into things like management, targeting establishment, or targeting spread of species. The last thing I just want to touch upon really briefly, I sort of quickly glossed over this idea that there's multiple ways we can quantify functional diversity. And there's this idea of functional richness, which is the total spread or the range, and functional uniqueness. And a lot of what I'm interested in over the last couple of years is trying to really dive into these different types of diversity, different ways we quantify biodiversity, to try to understand how different facets of biodiversity can help insulate systems against different disturbances, climate change, invasive species, and so on. So here's a nice little diagram showing how we can think about these different types of functional diversity. So this is work with Andrea Paz, who was also a postdoc at ETH with me. She's now a professor at University of Montreal. So the thought was to try to take this idea of functional diversity and look at these global patterns and try to link these global patterns back to things like invasion. So across the rows here, you have divergence, low and high divergence. Across the columns, you have richness, low and high richness. Now, richness, again, is just the spread. So if you look at this high richness category, these are both high richness because they have the maximum range of traits. Here, the colors just represent a trait. You could picture it like leaf size or something like this, or bark thickness. So there's just a single trait axis, and then we just order them on this trait axis. And these two groups, although they look very different, have the same min and max. Divergence represents clustering or uniqueness. And so low divergence is essentially settings where you have clustered species and you have gaps in trait space. So essentially like empty niches exist because their species are sort of redundant with each other, versus high divergence, high uniqueness, low clustering. Things are more uniformly spread across this niche space. So as we saw in this previous slide, they have slightly different roles. So functional richness, which is again the range of traits, that has the strongest sort of buffering effect against invasion. Functional divergence, uniqueness matters, but maybe not quite so much. What we can do is look at these global patterns to help us inform maybe where different areas might be susceptible to different types of invasion. When we do this, we see first off functional richness, this upper left panel, and also this black line on the plot. This just essentially tracks species richness. So it gives us the latitudinal gradient. You have maximum functional richness in the tropics. The idea is when you have 10,000 species across the landscape, you have just an enormous spread. Regardless of how clustered they are, you've probably sampled a huge range of traits. So the range of traits is maximally high here, and it just decreases as you move poleward. What I think is more interesting is this divergence or uniqueness of species. And Europe is a bit messy just due to sampling artifacts, but if we focus on the Americas, you can see very clearly that we see this inverse relationship, this hump-shaped pattern, and you can really see it clearly by the green on the right-hand panel, that uniqueness of species is highest in temperate ecosystems and actually lowest in the boreal and in the tropical ecosystems. And this is because tropical forests actually are made up of very few genera, right? They're essentially very, very clumped. You have 20,000 species in the Amazon basin, but they really come from very small range of trait space. So they occupy a very big spread, but they're clumped very tightly within them. So this gives us very, very different global patterns about functional richness. And the question is, how might this feed into things like invasion? So again, think of this lens. So here, functional richness, it shows the tropics should be very, very strongly buffered from invasion. And of course, that's exactly what we see globally. We have most of our invasion happens in northern latitudes. Native redundancy still plays a role, which suggests maybe the temperate systems might have some additional buffering that maybe we wouldn't expect based on richness. But it does suggest that really high latitude boreal forests, for example, are really, really susceptible to invasion from sort of all angles. They have no richness, they have no redundancy. They're both small ranges and also very clumped as well. We can do this a bit more quantitatively. So here we have a plot showing the 12 major biomes, and we have functional divergence and functional richness. We have these quadrants drawn sort of to match what we have up here. This dark green in the lower right, that's the tropical broadleaf forests. And so you can see very, very high functional richness, but actually incredibly low functional divergence, one of the most clustered biomes. And its level of clustering sort of matches what we see in boreal forests. So boreal forests are also very, very clustered, but also have very low richness. So they're sort of down here, low richness, low spread, and clustered. Tropical systems have a wide spread, but are very clustered. Temperate systems sort of fall somewhere up here in the middle on average, right? So they have sort of moderate range and some degree of clumping, but much, much less clumping than we otherwise see in tropical systems. So just by looking at these different facets of functional diversity, we can start to visualize how these systems are structured a little bit differently. What's also very cool is we can link this in with what we see in the invasion results. So here we have a plot where across the bottom we have temperature, and then we have what's called invasion strategy. So this was going back to Camille's work, whether the species that happened to invade, did they invade by being more dissimilar from the native community, or did they invade by being more similar to the native community? And what we see, not super pronounced, but there's a significant relationship, is this hump-shaped pattern, right? That when you're in very cold, very cold environments and very moist environments, you actually tend to invade by mimicking the system, by sort of being very similar to what's there. And this matches with what we see based on the functional composition. So in these settings, we see very strong clumps. And it suggests that to survive in these settings, you have to be in one of these clumps. There's no other way to survive, or else in the tropics, they would have exhibited these traits. So you have to mimic what's there to survive. And this is what we see playing out. The invasion happens by mimicry in these settings. Conversely, in more temperate systems between 10 and 20 degrees, this is where we know things have maybe slightly reduced trait range, but they have relatively high uniqueness. And here we see that things invade by being dissimilar. What this means is they're not invading into the interior of trait space, they're invading out on the edges. And this is exactly what we see. There's really nowhere to invade in this niche space. Like every possible niche internally is occupied. So the only way you can invade is being very different. So you invade on the outskirts. You know, you're a purple tree who manages to make it into the tropics or in temperate systems, and you can invade. So by looking at these global patterns, we can start to make sense of some of these relationships that we see and understand the mechanism of invasion. So not just spread versus establishment, but also trying to understand where species might invade by being more or less similar through this lens of functional diversity. So to sort of wrap this all up, the key sort of tenet of a lot of my work is that diversity, be it species, interspecific variation, functional diversity, really helps promote and sustain diversity in numerous ways. So when we look across these different forested ecosystems, for example, we know they all have different types of functional diversity, above ground, below ground. They have different levels of uniqueness, spread of different traits, and that all of these are going to give them slightly different ways that they can sort of inhibit against spread of invasive species, that can buffer against subsequent loss of species, but that this is going to be slightly different in these different environments. The one thing sort of to keep in mind is that human activity can ultimately overwhelm this in every case. So here we're looking at invasion strategy when you're far from a port. So when you're more than 500 kilometers from a port, we see this positive relationship. So you move from similar to dissimilar, and this time we're looking at just precipitation, not temperature. So we see this linear trend. However, when you get close to ports, we see an inverse relationship. So humans have the ability to essentially just completely destroy whatever the biological capacity is of that system. And even through enough attempted reestablishment of species, any sort of native buffering capacity can be destroyed and potentially even just inverted based on the species that are being introduced to these areas. So although there is this buffering effect of biodiversity, it's not separate from human activity. And enough human activity, unsurprisingly to all of us, I think, can overwhelm any sort of degree of biological capacity that these systems have to buffer against loss of species. But I still think there's sort of a powerful note here. Promoting diversity can prevent these negative feedback loops that would otherwise further reduce diversity. And so the challenge is if we lose a bit of diversity, we're going to be at risk of losing even more diversity, which puts us at risk of losing even more diversity. I think this is sort of promising because so much of what we think about through diversity is because it's like a commodity that can be linked to things like how much carbon can we store in this forest, how sort of many ecosystem services can we provide, what's the economic value. What this really shows is that diversity has a value separate from all of those things, that diversity in and of itself is integral to maintaining itself. So we get this sort of paradoxical result that maintaining biodiversity is critical for the maintenance of biodiversity. And maybe this isn't surprising, but also we can really show, whether it's experimental, theoretical, and scaling all the way up to the global scale, that diversity really helps support diversity and can help prevent subsequent losses in diversity in these systems. So I really think that I like to view it as a positive message that we now have the potential to protect biodiversity, and in doing so, we actually we sort of foster these ecosystems to be even more and more resilient just by focusing on biodiversity per se. And with that, I would like to thank all of the different places I've been where this work was done. A big thank to Andrea Paz and Camille Delvaux over the last few years, as well as my funding sources. Thank all of you for listening, and yeah, it'd be great to answer some questions.
Amazing.
Thank you, Dan.
Go ahead, Colin.
So I think we're gonna kick off a round of questions. So feel free, if you have a question, to pop it in the chat, and I will review them and ask them for y'all. And I'll say your name to credit, because that's important. So yeah, anything you want. And Dan, I might just kick it off with a question of my own to get us started.
Yeah, go for it.
First of all, that was awesome. You showed in so many different ways how, complexity or diversity in and of itself, allows the maintenance of diversity. I think it was super clear. A lot of it was through this lens of sort of resisting invasion. And I'd love to ask you the question of, like, if we flip that on its head and we thought about this in the context of restoration, right? So in a restoration context, we have some quote unquote degraded ecosystem. We're trying to reestablish some reference ecosystem. You can imagine situations. We always talk about this in tropical forest restoration. Like, we want to plant lots of species, not just 2 or 3 species. Yeah. And would these frameworks predict, like, the successful restoration should in theory be a function of the diversity in which you establish in the first place? We also think about this in human microbiome interventions and many other things.
Yeah, I mean, I think it's a great point. I think, people like Leland who are actually doing restoration through the lens of functional ecology, I think there's a lot of push towards not just planting diverse species anymore, but planting, to the extent we're using planting, I know that's a loaded term, but to the extent we're restoring groups of species, that we want to focus as much on their functional uniqueness, so their role in that ecosystem. And you can think we're sort of trying to do the same thing, that when you restore an ecosystem, you're going to have forces that allow it to build up, so succession, but you're also going to have some sort of destabilizing forces that are trying to pull it down. And so if we have the right combination of biodiversity, we can at least minimize that destabilizing force that's going to tug this thing down. So this is really why when we look at more diverse restoration, where they're not just trying to foster 1 or 2 species, but many, many different species, these tend to be more successful, maybe not initially, but over the long run, because we think we're like stacking blocks, and they have a much stronger foundation in that setting. So I think these sort of results really suggest that things like functional ecology have a huge role in restoration ecology, because we can start to determine, this is up to the individual practitioners, but what sorts of traits, what sort of niches, what sort of life history strategies can help give you these sort of complex relationships that prevent any one species from becoming monodominant, for example.
Awesome. Great. We have questions coming in. Yeah, it's really interesting though. Yeah, I mean, I think it's always been intuitive that, like, we think when doing restoration, whether thinking about like human gut microbiome restoration or, replanting a forest, right? Like, more species good. But I actually am not aware of a lot of solid theoretical grounding there. And so this feels like that lens. Kaylin, I see you asked a question, so you want to just go for it and I'll review these other ones.
Yeah, great talk, Dan. That was really interesting. My question is, do you know of or envision any invaders that really thrive in high biodiversity that where in low biodiversity be for competitors?
Yeah, I think like, again, that was all just looking at trees, but I think when we expand more broadly, we definitely see this. So a lot of like understory shrubs that are invasive, that they thrive in really close canopy environments. So you do often see invaders that can occupy this sort of niche that nothing else could occupy. And these can really thrive, particularly when there's nothing else left but that niche. So, really close candidates, quite dark in the understory, maybe not much locally. There's not many plants that can survive that. And this is where you can get this explosion of an invader. I don't know if that'd be sort of diversity per se, so much as just like a lot of things. I think diversity, that's a really good question. I can't think of anything. I think you might have to look to other trophic relationships, things like herbivory, these sorts of invasive species that would benefit from having a lot of different leaves to eat, or root pathogens, these types of things, I think would thrive in those settings. But when we're thinking more of a single trophic level of just trees or plants, I think it's a little hard to make a case that you'd get these big things. Just one last thing, sort of to go what Colin said, I think it ties in a little bit that I think we can get, like, if we're trying to do gut microbiome restoration, we sort of know more is better, in part because we're like trying to build a house of cards, but we're doing it just by throwing things in. And we sort of hope if we throw enough rocks in, we can like come out with a relatively high pile of rocks. But certainly there are, like, you think of the gut microbiome, there's viruses, there's bacteria. There are these things that get in and can just lay waste to that system. And those are like the true invaders. Again, I don't know how much they would benefit under diversity. I still think in all these settings, diversity of gut bacteria is going to be beneficial in general. But certainly these things can sort of ignore that altogether if they're strong enough.
That makes a lot of sense. Tessa, this is a great question, and I'm realizing it's probably just easier if you ask it yourself. So I'm going to ask you to unmute and chair. And thanks for coming.
Thank you. Hi, Danielle and Colin. Yes, I very interesting talk, and I like your fungal data, of course, a lot. I was wondering, I also work on fungal interactions at the moment, and I was wondering if you ever tried to bring positive interactions in, like something like cross-feeding, or like recent reviews were showing that these positive interactions may be even more relevant in the soil microbial communities. So I just wonder if you ever tried to make calculations or experiments.
Yeah, so the closest we did is in that system, we've started to do some work on higher order interactions, and we tried this a couple different ways. We actually had an experiment going in ETH, which is sort of half done. I have some results as well, and what we actually find is that so higher interaction, for those who aren't familiar, is, the species A might be species C, but when you add, or A might be B, but when you add C, that relationship could flip, or at least the rate at which something's competitive changes. So you add in a third species, and the relationship between those two changes. And a good example of this would be like prey switching. So you have a wolf eats a moose, but then you add an elk, and all of a sudden that moose gets a reprieve, right? Maybe it turns its attention to the elk. And so the closest we get in this is that in fungi, they can be sort of beneficial, right? They can use secondary byproducts. Even in those wood decay fungi, they can sort of grow over each other. In some of my work, we actually in the field looked at the proportion of positive relationships as a predictor. Essentially, how often do you see things coexisting together than you would expect by chance? And what we sort of find is that certainly there are higher order effects, these things that happen. So interactions flip fundamentally when you add in another species, and particularly things that deadlock together. If you have two species that draw, when you compete them, adding a third species can allow them to sort of start to move in unison. And so you can get these relationships where they sort of join forces, for lack of a less anthropomorphized term. But that's sort of the most we've seen in my system, in part just because I'm not doing like enzyme analysis. I'm not actually looking at the mechanism of relationships. I think more broadly, positive interactions are huge, and particularly, of course, for trees and fungi, this is going to be a really massive relationship. But I'm not really certain how this affects diversity. In fact, there's a lot of theoretical results showing that positive relationships are actually quite destabilizing in a lot of systems. And you can think of this through the lens of like positive feedback. So if you think of alternate stable states in forests, right, this is driven by a positive feedback. And you have this positive feedback that allows things to become very, very dominant because they partner up together. And what we're left with in forests is sort of these two different states. We don't have a lot of mixing of different forest types because you have AM and EM, but you don't have a lot in the middle. And that's driven by these positive feedbacks. So positive feedbacks or positive relationships can have this sort of it's a double-edged sword where it can drive things to be very unstable because things can become so dominant and powerful because they're being supported by another species.
Thanks a lot.
Awesome. Awesome. I think we got one more question from Dr. Ann Reho.
Hi, thank you so much for your talk. I really enjoyed it. And yeah, it brought me back to, paleo days when I was looking at the map of North America, and I was just seeing right away the Great Plains and the Prairie Peninsula. And I know, we have woody encroachment in those areas. And so how much does historical context matter when you're thinking about, the probability of invasion?
Yeah, this is huge. And I think this was like part of half of what we spent time with reviewers on in that paper. Because yeah, there's a big issue. I mean, a great example in Central Western America is, I can't remember the species now, but they planted it to create like fence posts. It was like the main tree species for fence posts. And it's all over the Western US now because it was essentially planted. This is a big challenge. I think one of the values here is that we can take a sort of management view of this and just say, this is some sort of introduction, right? And whether that introduction is because people want to grow these things, whether it's accidental. We also do sensitivity analysis in this, or remove any tree that's non-native, and we still see these same results. I don't know if you noticed, I'm not sharing my slides, but one of this distance supports like hooked back up at the end. I don't know if you saw that. There was like, you get far away and all of a sudden it scoots back up. And that's exactly what you're talking about, that you have these clusters of really weird artifacts because of sort of this weird historical contingency of how a specific tree was used in a specific environment. So I think there's a real challenge, and part of it would be distinguishing truly invasive from non-native. And we didn't make that distinction for sort of political reasons. We just say if it's non-native, but sort of what you're getting at is this idea of invasive, which is meat that's gotten there and is now spread out of control, versus a non-native where we planted it, no one cares about it, it's fine, it grows there. But I think particularly for trees, this is huge because most trees that are non-native most of them were brought there. It's relatively easy to control tree invasion compared to things like plants. You can just cut them down. Yeah, so part of it was, since we're looking at trees, we're sort of dealing with all the same system in general. But it certainly is important. It can lead to these really weird spatial artifacts in these data sets.
Yeah, that totally makes sense. Thank you.
Well, this is really exciting. I think, one of the things we call out a lot, so of course we work on restoring below ground biodiversity. And part of that is like, is it going to establish? One thing we take a lot of inspiration from is work in medical microbiology, where, some of the treatment for treatment resistant C. diff, so antibiotics aren't working, like this sickness you get when one microbe takes over, it makes you very sick, is a fecal transplant. And people have made reduced community treatments as well. They're like, okay, we think these indicator taxa in the fecal transplants are really driving this effect. Let's add them as a pill. Let's add 1, let's add 2, let's add 5 species, because it's really hard to grow complex communities in a bioreactor and scale them up. They either don't work or they don't work nearly as well as the fecal transplant in all its complexity. And I think there's emerging consensus in the medical microbiology side that it is something about diversity in and of itself. And I think we're seeing this thread emerge on lots of different sides of the research in lots of different communities. And so it's really wonderful to see these results and see you build this narrative around them, which is really nice. I'm impressed.
Yeah, I mean, I just want to say, I think that's very, very much like, thinking of inoculating soils, like, right? Intact soil is going to be better than a single fungus, almost certainly without a doubt, because you want to actually have, like, we don't necessarily know what matters, and it's this massive complex web that's necessary versus just 4 or 5 species. So I think it's still challenging, because how do you, if like you have soil inoculum, that's good, but fecal transplants are a little harder to sell to the public. So I think trying to find ways we can build these synthetic communities that really we can culture in high replication with hundreds or thousands of species that can coexist. I think that's really at that emerging frontier of how we can start to address these challenges.
I love that. That's how we're ending.
Yeah, that was the pitch.
All right. Well, thank you everyone for coming. Thank you from everyone, Team Funga, everyone from outside. We're glad you came. One last hand for Dr. Dan Maynard and all his expertise. And yeah, we'll be continuing this. We'll repeat this next quarter. So looking forward to seeing some of you again. And Dan, I'm lucky that I get to talk to you in another Zoom room very soon.
Thanks, everyone. It was really fun. If you have any questions or anything, shoot me an email. I'd love to chat more. Yeah, it was a real pleasure.
Thank
Bye,
you all.
everyone.
Have a good week.